Um, Ma'am, the last thing we discussed in the class is diffusion and osmosis.
Osmosis.
Yes, ma'am.
Okay. Okay, dear. Switch on your video. Let's start today's
Okay, ma'am.
class. All right. Now moving forward. So we discussed about the osmosis and we have discussed about the diffusion, right? Now,
Yes,
depending upon the osmosis, now see, the osmosis can be, you know, where it can be utilized. For that, you have to understand the solution. See, a solution is made up of two things. One is the solute, and the next thing is the solvent. For example, if we talk about sugar solution, okay. So in the sugar solution, there is like the sugar which is the solute, and the thing that you're dissolving it into, like the water or the soda, that becomes the solvent. Okay, so the solvent is always the liquid, whereas the solute is always the thing that you are dissolving. Now, these solutions, they can be of three types. Okay, hypertonic solution, hypotonic solution, and isotonic solution. Now, hypertonic solution, let's see, in these cases, what they're going to ask you is that what will happen to the cell if you keep it in a hypertonic solution? What will happen to the cell if you keep it in a hypotonic solution? And what will happen to the cell if you keep it in a isotonic solution? This is the question. Okay. Now, if we are talking about the hypertonic, the first case, here you can see that, in the external environment, there is more solute, so the water will be less. For example, if you take half a glass of water, or let's just say you took a glass and you filled it up with sugar. Now, now you're filling it up with water. So, do you think a lot of water will get into it?
uh, no, ma'am.
Why did you have to think about it so much? Obviously, it will not. If you take a glass, fill it up with sugar. Now you try to add water, only this tiny bit amount of water will end, right? So that is the case of hypertonic solution, when the solute concentration will be high and the water concentration will be low. So now if you take a cell and you put it in this solution, the cell has more water as compared to the outside. So now the water from the cell will move outside the cell. And now since the water is moving outside the cell will shrink up. So this is called as what? The hypertonic solution.
Hypertonic.
And if you keep a cell in a hypertonic solution, the cell will shrink. Then
Yes.
the next case is hypotonic solution. So in this case, the outside solution has more water, less solute. Now, if you keep the cell in it, since it has more water as compared to the cell, now the water from the solution or the external environment, it will go inside the cell. And this is called, like, it will cause the cell to swell up. The cell is going to swell because it's getting water inside it. So this is what will happen. So what will happen to the cell? If we put it in a hypotonic solution, it will swell up. If we put it in a hypertonic tonic solution, it will shrink. Now, here, since the water is either moving outside the cell or into the cell, so this is osmosis only, right? The movement of
Yes.
water. Now, the movement of water that is taking place outside the cell, we call it as exoosmosis. Exo means outside. And the water that is moving inside the cell, we call it as endoosmosis. Clear?
Yes, ma'am.
Now, isotonic is when the external environment has the same amount of water as the internal environment of the cell. So, there will be no change in the cell if you put it in an isotonic solution. Clear?
Yes, ma'am.
Hmm. Now, moving forward, so like they'll ask you this thing, that what will happen to the cell if you put it in a isotonic, hypertonic, hypotonic. So, is that clear to you?
Yes, ma'am.
See, and the process where the water was moving out of the cell, that was exoosmosis. And apart from that, it is also called as plasmolysis. It's the same thing, which is moving the like movement of the water outside. Clear?
Yes, ma'am.
All right. Now, now this whole entire property of osmosis, diffusion, it is the property of the plasma membrane. And you know that the plasma membrane is very flexible. But the question comes along that what is the plasma membrane made up of? So the plasma membrane, it is made up of two things. The first one is the lipid and the second one is the protein. Now if you look at over here, so talking about plasmolysis, so whenever a living plant cell loses the water from it through the osmosis process, right? Which osmosis? Will it be exoosmosis or endoosmosis?
Exhaust.
Yes, exoosmosis. So when the water will be moving outside the cell, right, which is causing the cell to shrink or contract. So that is called as plasmolysis, this phenomenon. Okay. So plasmolysis, exosmosis is almost the same thing. Now another term comes along, that is endocytosis. Now endocytosis, endo means inside, right? Now it is the process in which, like you might have heard about the organism amoeba. So amoeba, you can say it procures its food, meaning it eats with the help of this endocytosis process. How? For example, if there is a food near the amoeba, what does it do? It draws out its pseudopodia like this. It takes out the pseudopodia, circles the food, take it inside. This whole entire process is called as endocytosis. So here, what happens is that the food is engulfed inside from the external environment. It takes place as the cell membrane is highly flexible. That is why it is possible. If the cell membrane hadn't been so flexible, none of it would have been possible at all. So now what will happen is that so basically when we talk about the endocytosis, so here the food molecule is taken inside the body of the amoeba. Now, unicellular organisms such as amoeba, it acquires its food, meaning it gets its food through this process. Clear? Endocytosis is
Yes,
clear to you?
yes, ma'am.
Very good. Now, moving forward, let us see. So, another name for endocytosis is ingestion. Okay, like the another name you can give to it will be ingestion, right? Taking in of the food. And it takes place mostly in the case of amoeba with the help of pseudopodia. What is pseudopodia? The false feet, with the help of which the organism is able to take in the food inside. Hmm? So, from where? So, from the external environment. That is what we are terming as endocytosis over here. Any doubts?
So,
After this, the plasma membrane is now completed. Now we move on to another layer. Now, if you remember, the plasma membrane is the layer of the cell, and outer to the plasma membrane, another layer is present, which is called as the cell wall. Now, the cell wall, it is basically present in the plants, it's present in the this thing, bacteria, and it's present in the fungus. But is it present in animals?
no, ma'am.
No, 100% not. So basically, what happens is that the cell wall surrounds the plasma membrane. It is a very rigid layer that will be surrounding the plasma membrane. Now, this one is not present in the animal. Now, the cell wall in the plants, it is made up of a compound which is called as cellulose. So, we say that plants, they have a cellulosic cell wall. Cellulosic means that it is made up of cellulose. Now, furthermore, what happens is that in the case of animals, obviously, they do not have any cell wall. But We saw that a fungus and a bacterium, they have a cell wall. Now the cell wall of the fungus, it is made up of a compound which is called as chitin. Whereas in the case of bacteria, the cell wall is made up of a compound called as peptidoglycan. So plants, fungi and bacteria, they have the cell wall present, but their cell walls are made up of different components. For plant, it is made up of cellulose. For fungus, it is made up of chitin, and for bacteria, it is made up of peptidoglycan. Any doubts up until now? Hmm. All right, now moving forward. Now, when we talk about the cell wall, what is its purpose? Why is it there? Right? What is the need? So, the cell wall, first of all, it protects or it prevents the cell from bursting. It will not, like we saw how the plant cell, we were keeping it in a hypertonic solution, hypotonic, it was shrinking, it was swelling up. But was it bursting? No, wasn't bursting. Why wasn't it bursting? Because there is the presence of cell wall, and the cell wall will not allow the cell to burst or collapse. Apart from the plants, the organisms such as bacteria and the fungi, they have a cell wall. Bacterial cell wall is made up of peptidoglycan, and the fungal cell wall is made up of chitin. Clear?
Yes, ma'am.
So up until now, do you have any doubts?
No, ma'am.
Plasma membrane is clear.
Yes, ma'am.
Different types of solutions are clear.
Yes, ma'am.
And cell wall is clear.
Yes, ma'am.
Okay, very good. Now see, moving forward, we will be talking about the nucleus. Now inside the cell, there is this structure which is called as the nucleus. You can call it as the control house of the cell. You can call it as the brain of the cell. Why? Because it will be controlling everything related to the cell. Hmm? Now, it, like the nucleus, it is the most darkly stained region of the cell. It is that part of the cell that gets stained a lot. Okay, so why, and can you tell me why do we need to stain the cell?
Ma'am, can you repeat again? What did you say?
Why do we need to stain the cell?
Uh, no, sir, ma'am.
See, what happens is that the cells and the cell organelles that are there, they are mostly colorless. Okay, they are mostly, they do not have any color present on them. So when we want to see them under the microscope, we have to give them a color so that we will be able to see them. So for that, we stain the cells, we color them with the help of certain stain so that we can visualize them, we can see them clearly. So what are the common stains? We use iodine solution, we use saffron in solution, methylene blue solution, Janus green. There are many different kinds of dyes that we use. Now, obviously, if you have ever colored a cloth, you might have noticed. Now, why would you color a cloth? But still, you might know that there are some things that can take up the stain a lot, that becomes very dark in color. But some things, even if you put color and color, they'll not take up a lot of stain. So nucleus is that part of the cell which is going to take up a lot of stain. So it is the darkly stained region of the cell, very dark. So when you see under a microscope and you see several cell organelles, amongst that, if you see a part which is very darkly stained, so you'll already understand that it is the nucleus, right? Now the nucleus, it is surrounded by two layers. Okay, so it has a double layered structure which consists of an outer layer and an inner layer. So the outer layer of the nucleus and the inner layer makes it a double membranous structure. And the membrane of the nucleus is called as nuclear membrane. So it's like a protective layer surrounding the nucleus. Now tell me one thing. Will the prokaryotes have the nuclear membrane?
No, ma'am.
Why?
Ma'am, prokaryotic cells does not have nucleus and nuclear membrane in them.
Hmm. Very good, exactly. The prokaryotic cells, they do not have a nucleus. How will they have a nuclear membrane? Because any kind of membrane bound structure is not present in them, right?
Yes,
Now,
ma'am.
this nuclear membrane, it contains a lot of pores in it for the transfer of materials into and out of the cell. So if this is the membrane, so it is surrounded by a lot of pores like this. And these pores that are present in the nuclear membrane, we call them as nuclear pores. Why are they there? They are there for the transfer of materials from the inside to the outside and from the outside to the inside of the cell. Is it clear?
Yes,
Any
ma'am.
doubts?
No, ma'am.
Okay. Now, you might have heard about DNA, right?
Yes, ma'am.
So DNA is that genetic material in us that makes us who we are. Now, the DNA is present inside the nucleus. That is why the nucleus is very powerful. Now, with if we talk about the DNA, we
Ma'am,
have
could
to
you repeat
talk
again?
the DNA. It is present inside the nucleus. That makes the nucleus very very powerful. Okay, it makes the nucleus sort of like a control of every single thing. So when we talk about the DNA, we have to talk about a thing called as a chromosome. What is a chromosome? So if you look at the microscopic image and inside the nucleus, you will find a rod shaped structure. Okay. And these rod shaped structures are called as the chromosomes. Now the chromosomes, they contain the information of inheritance. Meaning, what is inheritance? Like if you think about it, like if you compare yourself to your parents, you might have some features of your mother, some features of your father, or some a combination of them both, isn't it? So where is it coming from? It's because you've come from your parents, and while you've come from your parents, you have been passed on the gene or the DNA from your parents. And this process wherein next generation gets the DNA or the information from the previous generation, meaning from the parents, we call it as inheritance. So when we talk about inheritance, it is the passing of traits or the passing of characters or features from the parents to the offspring, that is the children, through the process of sexual reproduction. Is it clear?
Yes, ma'am.
So I'm saying that the chromosomes, they contain the information of inheritance, and that is the DNA. So inside the nucleus, the chromosomes are present, and the chromosomes, these are the carriers of DNA. They carry the DNA on them. So the DNA is present on the chromosome. It's just not there. It's present on the chromosome. Now talking about what is sexual reproduction. So it is the type of reproduction where two parents are involved and it involves the fusion of gametes. So this is like a side note to understand what is sexual reproduction. Now the DNA we are talking about, the DNA, first of all, you should know the full form of it. It stands for deoxyribonucleic acid, right? It is our genetic material. It makes us who we are. DNA is present everywhere in our body, in our saliva, in our hair, everywhere. Hmm? Now, the chromosomes, they contain the information of inheritance of characters from the parents to the offspring, and it is contained in the form of DNA molecule. So if anyone would ask you that what is the chromosome made up of? What is it made up of? So your answer should be that it is made up of DNA and proteins. Two things make up the chromosome. One is the DNA and the other is the protein. Is it clear?
Yes.
Hmm. Now the part of the DNA that transfers the information from the parents to the offspring, that part of the DNA is called as the gene. Okay, so the part of the DNA which will be sort of like a functional unit of DNA, we will call it as the gene. Okay,
Ma'am.
now moving forward, always remember that whenever a cell is not dividing, you know that the cells divide to form new cells. So whenever the cell is not dividing, so in that case you cannot easily see the cell. The DNA will be looking like this. You cannot see it clearly. Okay. So this tangled mass that I just drew or scribbled, it is called as a chromatin. So whenever the cell is not dividing, the DNA is present as a part of a chromatin material and it is entangled. Okay. Entangled, you understand? It's like ulja hua, not free.
I tangle, ma'am.
Hmm?
It is all tangled up.
Yeah, it is all tangled up. So now inside the cell there is the nucleus. Inside the nucleus we have the chromatin material. Now this was when the cell was not dividing. Now as soon as the cell will start to divide or decide to divide, then this tangled stuff which was there, the DNA, will start getting organized. It will start getting untangled and it will get organized into the structure called as the chromosome. And we have already read about the chromosome, that it is the rod shaped structure, right?
Yes.
Now remember that one chromosome is made up of two chromatids. One chromosome is made up of two chromatids and they are joined together in the center by the structure called as centromere. So for example, if this is one chromatid and this is another chromatid, TID, right? And when they join together like this in the center, then it becomes a chromosome. And what is the structure that joins them together? It is the centromere. So remember it like this: center, centromere. Clear? So one chromatid plus one chromatid is equal to
Uh, sister chromatid.
hmm?
No, nothing.
No,
Centromere.
no. See, I'm saying one chromatid plus one chromatid is equal to
Uh, it should be sister centromere. No, it should be centromere.
no. Centromere is joining them. My question is, if you add one chromatid to another chromatid, what will it make?
Will make sister chromatid.
Sister, brother, don't do that. See, one chromatid plus one chromatid is equal to one chromosome, isn't it? So simple, it's not complicated. One chromosome is made up of two chromatids and they are joined together at the center by the structure called as the centromere. Now these two chromatids, these are the sister chromatids. Okay. How do you know sister chromatids? They've taught you in the school.
Yes, ma'am.
So any doubts up until now?
Ma'am.
Also, one more thing, dear. While I'm teaching, if there is anything extra that has been taught in your school, do let me know so that I can also teach that to you.
Okay, ma'am.
Okay, maybe something that might not be there in my notes and you have learned it, just like how you said sister chromatid. It's very new and it's not there in class 9, but they have taught you. So if there's anything, you do let me know. Okay.
Ma'am, it's actually that school we are not following an entire book. We are following an advanced guide. So
Uh,
because of this, our guide contains past and question of class 10 and 11.
okay, so they are teaching you a higher level, like they are using a higher level book to teach this chapter.
Yes, ma'am.
Okay, okay. So yeah, so that is what I'm saying. Whichever book they are using, do you have that book?
Yes, ma'am.
Can you
Hey,
send the
so
PDF if possible?
yes, ma'am.
Okay, whenever like after the class, you send that PDF to me. Okay, like just send it to the team. Ask them to send it to me. Okay. Hmm. All right. So now the nucleus, like present inside the nucleus, is the nucleoplasm. It's like a fluid-like substance that is present inside the nucleus. Now let us look at this thing. Chromatin. It is the tangled state of DNA whenever the cell is not at all dividing. Now looking at the functions of the nucleus. Now the nucleus, it plays a very central role in cellular reproduction, meaning cell division. Without nucleus, cell division cannot happen. And it is the process, like this cell division process, it's the process by which a single cell will divide and form two new daughter cells. So this process will be taking place because of the nucleus. Other than that, the nucleus controls the functioning of all the cell organelles. It regulates the division of the cell, and it contains the chromosome and the DNA for inheritance. The functions of the nucleus are clear to you?
Yes, ma'am.
Hmm. Now, see, the next thing is actually the cytoplasm. No, before that, let us see the differences. But first of all, you tell me, up until now, do you have any doubts?
No, ma'am.
Okay. Can you tell me what is osmosis?
Osmosis is a process from where water low concentration will move to a higher concentration.
No, from high to low.
Oh yes, ma'am. The higher to low
Always
concentration.
high to low, never from low to high.
Yes, ma'am.
Hmm. Yeah. So it is the movement of water. So does the water follows the law of diffusion?
No, ma'am.
Yes, it does.
Ma'am,
Oh,
I don't know the law of it.
the law of diffusion says that anything will move from a region of its higher concentration to the region of its lower concentration.
Oh yes, ma'am. Then it will.
Hmm?
Oh yes, ma'am. Then it will follow.
Yeah, yes, 100% it will follow the law of diffusion. Okay. What are the two types of osmosis?
And osmosis and exosmosis.
Okay, very good. Okay, what is the difference? What is the meaning of plasmolysis?
Plasmapheresis is another name of exosmosis. Exosmosis.
Very good. Okay, can you tell me what will happen to the cell if we place it in a hypertonic solution?
When you place a cell in a hypertonic solution, then the cell will shrink up due to
Hmm,
osmosis.
very good. Due to endosmosis. Very good. And what will happen to the cell if you put it in the hypotonic solution?
When you put a cell into hypertonic solution, the cell will swell up due to exosmosis.
Due to
And osmosis. And osmosis.
endosmosis. Okay. All right. Okay. And what will happen to the cell if you put it in an isotonic solution?
No reaction will occur.
The cell will remain the same. Now we
Yes.
saw that the cell is shrinking also, it is swelling also. So why is the cell not being destroyed?
Ma'am, because of cell wall.
Very good. And in which organisms are the cell wall present?
In animal and bacteria cell.
In
In plant and bacteria cell.
okay, plants and bacteria. Is there anyone else where it is present?
No, ma'am.
Fungus. Just 1 second,
Oh
yes.
yes, our fungus.
Okay, just 1 second. What? See. So yes, what was I saying? So it is also present in the fungus. Now, can you tell me what is the cell wall of plant made up of?
The cellular plant is made up of cellulose,
Cellulose. The cell wall of bacteria,
uh, nurture.
peptidoglycan. Okay, peptidoglycan. And the cell wall of fungus,
It's protein.
chitin.
Yes, ma'am.
Okay, chitin. Chitin. If you have trouble remembering chitin, you can say chitin also to remember the spelling. Okay, just to remember the spelling. Okay.
Okay, ma'am.
All right. Now moving forward, so we have discussed about the nucleus. Okay, tell me how many membrane is the nucleus surrounded with?
The nucleus is surrounded with two membranes.
Two membranes, outer membrane and inner membrane, right? And the
Yes.
membranes of the nucleus are called as
Uh, it is known as nuclear membrane.
nuclear membrane. Okay, what are nuclear pores?
nuclear code of codes that are present on the plasma membrane for in and out for genetic material to switch in and out.
Not genetic material. So certain materials to transfer. Genetic material doesn't go in and out through that. Okay. Okay, very good. All right. Now, can you tell me what is the basic difference between a prokaryote and a eukaryote?
A prokaryotic cell does not have a nucleus, and eukaryotic cell does contain nucleus.
Hmm. Other than that, let us see more differences. So the prokaryotic cell, they are always smaller in size. They're very smaller and as compared to the eukaryotic cell. In them, nuclear membrane is obviously not present. Any kind of membrane bound cell organelle is not present. Whereas in the eukaryotic cell, it is present. Right? And the example of the prokaryotic cell is bacteria, whereas the example of a eukaryotic cell, plants, animals, and fungus. Now moving forward, talking about cytoplasm. Now, cytoplasm is that area of the cell which will stain the lightest. So, if you are looking at a microscopic image, like if you're looking through the microscope and you see a structure that is very lightly stained, so you can understand that, okay, this structure is the cytoplasm. Okay, very lightly it takes up the stain. Now, cytoplasm, it is the fluid that is present inside the plasma membrane. Now, the nucleus and the cell organelles, like every single component of the cell. For example, if this is a cell, inside the cell, there is the nucleus. There is mitochondria, ribosomes, right? Like endoplasmic reticulum. All of these we will be studying, but these are the cell organelles. Now, all of these structures, they are present embedded in this fluid-like structure that is present all around in the cell, which is called as the cytoplasm. So the nucleus, the cell organelles, all of them are present right in the cytoplasm. And that cytoplasm is very commonly called as the site or the arena of all the cellular activities. Meaning every single activity of the cell, it actually happens or takes place inside the cytoplasm. Is it clear?
Yes, ma'am.
Any doubts?
No, ma'am.
Well, has this chapter been completed in your school?
No, no, not yet.
Not yet. Okay.
For in
Which
my
topic is
school,
going on?
we are discussing Golgi apparatus.
Golgi apparatus. Okay. Yeah, what were you saying?
I knew I was saying the topic.
Oh, you were telling the topic only. Okay. And for your test, that is in May, this whole entire chapter is coming, right?
Yes, ma'am.
All right. Now, can you tell me which structure or which part of the cell is common to both prokaryotes as well as eukaryotes?
Ma'am, ribosomes.
Ribosome, plasma membrane, right? Very common. And if there is a plant cell and a prokaryote, so cell wall is also common. So now let us look at it. So we have I told you about plasmolysis and endocytosis?
Yes, ma'am.
Now the cytoplasm. So it is the fluid that is present within a cell between the cell membrane and the nucleus, and all the cell organelles are present in that cytoplasm. It is the medium in which every single reaction of the cell, meaning cellular reaction, will be taking place in that area only. Talking about the nucleus, we call it as the brain of the cell or the control house of the cell, and it is responsible for the regulation of every single activity of the cell. Without this, the cell will not perform correctly. Other than that, there are other structures that are present inside the nucleus, which is the nucleolus and chromatin. These are extra structures that are present. Nucleolus is like a circular structure present inside the nucleus. It is responsible for forming the ribosome. Other than that, just like how the cell has a cytoplasm, same way the nucleus has the cytoplasm of its own, which is called as the nucleoplasm. So the cytoplasm or the fluid of the nucleus, we call it as nucleoplasm. Now, the nucleoplasm contains the chromosome, and the chromosomes, they contain the DNA. And the functional unit of DNA is called as what? It is called as the gene. Moving forward, whenever the cell is not dividing, the DNA is present in the form of chromatin. And when the cell decides to divide, the chromatin starts to get condensed or organized to form the chromosome. And the chromosome, these are the rod-like structures that are made up of DNA, and they are made up of proteins. And they are very important as far as the cell nuclear division is concerned. Other than that, the nucleus, it regulates the cell division, meaning from one cell, when multiple cells are going to be formed, it will happen because of nucleus, right? And it carries the heredity information, meaning that inheritance information that will be passed on from one generation to another generation. This is contained by the nucleus. So nucleus is clear to you now?
Yes, ma'am.
Okay, very good. Then comes the cell wall. We have already seen that it is a very rigid layer that surrounds the plasma membrane, right? It provides the structural strength and it provides rigidity to the plants. And it is present only in the case of plant cell. It is non-living, sorry, not only in the case of plant cell. It is also present in bacteria and fungus also. Okay, you don't write only. But it is absolutely absent in the case of animals. It gives a proper shape to the cell and it doesn't let the cell to burst or to collapse. Now we have also seen the differences that are there in between the prokaryotes and the eukaryotes. Any doubts up until now?
Ma'am, what is the meaning of rigid?
Rigid. Rigid means something that doesn't bend so easily. Rigid means stiff. Like if you try to bend a pen, it won't. So it's like rigid. But if you try to bend the paper, it will. So that's flexible. Got it?
Okay,
That
ma'am.
way.
Yes.
Now, the next thing that we are going to see are the cell organelles. So now, inside the cell, okay, inside the cell, there are specialized cellular structures. Okay, these are the specialized cellular structures that are responsible for carrying out different functions. Okay, specialized cellular structures. Let us see the most important cell organelles. We have mitochondria, Golgi apparatus, endoplasmic reticulum. There is plastids, but only in the case of plants. And we also have the lysosomes and the vacuoles. These are some of the important cell organelles. What is the use of the cell organelle? They are responsible for carrying out crucial functions of the cell. The most important things that takes place, like imagine like inside the cell, because of the presence of this cell organelle, the function or the work is divided. So it's like a ribosome. They're like, no, we'll not do anything else. We'll do protein synthesis. Now, the rest of the cell organelles, they are not worried about protein synthesis. Okay, so it will be the function of the ribosome, whereas the other parts, they will have some other function. So the cell organelles, these are specialized cellular structures that are responsible for carrying out crucial functions in the cell. Clear?
Yes, ma'am.
Any doubts?
No, ma'am.
Will the cell organelle be present in the prokaryotes?
Some of them, ma'am.
Yes, very good. Which one? In general, which one? Not the example. Tell me which one.
Lysosome,
Hmm.
plastids, ribosomes, and Golgi apparatus.
Plastids are not present. Plastids are not present in the prokaryotes.
Ma'am, not plastids, mitochondria.
Oh, mitochondria, definitely not. Mitochondria, plastid, they are surrounded by two membranes. Prokaryotes can't handle it. Ribosomes are present, vacuoles are present. Okay, vacuoles are also present mostly, and lysosomes, meaning all those structures that do not have a membrane, they will only be present. Rest all can't be present. Clear?
Yes, ma'am.
So the first cell organelle that we are going to discuss is the endoplasmic reticulum. Endo means inside. Plasmic refers to the plasma, right? The plasmic area. And the reticulum means a structure that is mesh-like or net-like. So it is a large network of membrane-bound tubules and sheets. It can be round, it can be long, or it can be oblong, like slightly elongated. Now, if you see a picture of the cell in which it is showing the endoplasmic reticulum, so always remember that the endoplasmic reticulum sort of gets continued from the nucleus. So it's like a continuation that takes place from the nucleus. Okay. Now, the structure of the endoplasmic reticulum, it is almost similar to the structure of the plasma membrane. Endoplasmic reticulum, depending upon whether or not ribosomes are present on it. The endoplasmic reticulum is of two types. Smooth endoplasmic reticulum, when ribosomes are absent on them. So it gives a very smooth appearance. And whenever the ribosomes are present on them, so it gives a very rough appearance, and that is called as the rough endoplasmic reticulum. Right? So smooth endoplasmic reticulum and rough endoplasmic reticulum, these are the two kinds of endoplasmic reticulum. Now, why ribosomes are attached? Why not anything else? Because ribosomes, these are those structures that are responsible for the synthesis of proteins. So it means that if the endoplasmic reticulum, if it is having a ribosome present on it, then it will be responsible for protein synthesis. It will be synthesizing the protein. Whereas if the smooth endoplasmic reticulum, if it doesn't have the ribosome present on it, then it will not synthesize the protein, right?
Yes, ma'am.
Hmm. Now see, we say that the ribosomes, they are present in prokaryotes and eukaryotes both. It's just that in them also there is a difference. The ribosomes of the prokaryotic organism, it is of 70S type, whereas the ribosome of the eukaryotic organism, it is of 80S type. S is the sedimentation. So even though they have this ribosome present in both, but it is of two different types in both of them. The superior kind, the better kind is found in the eukaryotes, whereas the inferior, not inferior, but the lower version is found in the prokaryotes. Clear?
Yes, ma'am.
So if I say, what will be the function of rough endoplasmic reticulum? What will your answer be?
Uh, my answer would be I forgot again.
Rough endoplasmic reticulum, it has the ribosomes present on it. So it will synthesize protein. And
Yes, ma'am.
smooth endoplasmic reticulum, it doesn't have the ribosome present on it. So it will not synthesize the protein. But what will it be its function? So what is the
It will
function? Huh.
detoxification.
Yes, very good. So it is responsible for the manufacturing of fat and lipid molecule. Detoxification, I think that is the function of Golgi apparatus. No, no,
No,
no,
ma'am.
no, no, no. Yes, yes. Smooth endoplasmic reticulum. Yes, and it is important for detoxification of drugs. They are teaching you class 11th bio. They're teaching you, I think they're teaching you from class 11 because this is not there in 9th. It's not there in 9th. What book are they using? Can you tell me the name of the book?
I don't know the name, ma'am. It's not written here.
It's not written.
I know, ma'am. It's not written here.
Okay, your school is using some sort of these supplicating books. Okay, you do send me the PDF if it is available. Okay, or the picture. All right.
Okay, ma'am.
So in general, what is the function of the endoplasmic reticulum? So the main function will be packaging. So it will synthesize, for example, if the rough endoplasmic reticulum, it is synthesizing protein. So those proteins have to be transported. So after synthesizing the protein, they will also sort of package it for the transportation. And they will also help in the transportation of that. Whereas the smooth endoplasmic reticulum, since they are responsible for the manufacturing of the fat molecules or the detoxification of drug, so they will do that, but they will not synthesize the proteins. Clear?
Yes, ma'am.
Okay, just give me 1 second. I'm connecting my laptop to the charge.
Okay, ma'am.
Okay. Now, moving forward, so we have seen the functions of the endoplasmic reticulum and everything that is there to know about them. Do you have any doubts up until now?
Ma'am.
Okay, moving forward. Now, when we talk about this thing, that is the membrane biogenesis. Now, membrane biogenesis, it is the process in which the cell membrane or the plasma membrane, they are formed with the help of proteins and lipids. The whole entire process through which the membrane formation will take place is called as membrane biogenesis. Clear?
Yes, ma'am.
Hmm. Ultimately, let us see the whole entire process of the endoplasmic reticulum. So it will be responsible for the transportation of cellular materials between the cytoplasm and the nucleus. It functions like the cytoplasmic framework, meaning it gives the cell or the cytoplasm sort of like a structure, okay, a particular structure. And apart from that, it also provides a surface for biochemical activities to be taking place. So these are all the different functions. Yes, I have written here also that smooth endoplasmic reticulum, it is also responsible for detoxification of poisons and drugs in the case of vertebrates. Okay. So
Okay, ma'am.
yeah. So this is all there is to know about the endoplasmic reticulum. Now tell me if you have any doubts.
No, ma'am.
Okay. Now the next cell organelle is the Golgi.
Ma'am,
Yes.
I think we skipped some parts of endoplasmic reticulum.
Tell me which parts?
Uh, like in my book, they give him detailed functions and like the detailed parts like cisternae, vesicles, and tubulus, and very detailed functions.
They've given detailed functions for that. It's
Yes,
not
ma'am.
there in your
Very.
syllabus. See, you have to send me this book. It's very important. Okay. Do send me this. Either you make a PDF or if the PDF is already available, you send it to me. Okay.
Okay, ma'am.
It's important because I'll use that to teach you only. See, cisternae, tubules, and vesicles. These are the structures that the endoplasmic reticulum is made up of. Right? These are the structures that provides them like the net-like structure that comes along. It's because of the cisternae, and cisternae are what? These are basically membrane infoldings, right? It is the membrane infoldings that provides the surface area also to the endoplasmic reticulum and a structure also, the network-like structure. Okay. Now, tubules and the vesicles, these are the secretory parts of the endoplasmic reticulum. So, if the endoplasmic reticulum are going to synthesize the proteins, so they will be secreting it out with the help of the tubules and the vesicles. Got it?
Yes, ma'am.
And membrane biogenesis is clear?
Yes, ma'am.
Okay. Then comes the functions, that is also done. Are there any more points in the functions of endoplasmic reticulum?
Yes, ma'am. They have given like five functions here, and every function has like very detailed scientific names, and like it's explaining in very details.
I'll see to that also. See, the basic function is this. Okay. What are the basic functions? The transportation of the cellular material between nucleus and cytoplasm. That is one of the function. It is responsible for providing the framework to the cytoplasm or framework to the cell. And it also provides that surface area for the biochemical activities. And the smooth endoplasmic reticulum, we already saw that it is responsible for the detoxification process, but this happens in the higher organisms, which are called as vertebrates. Okay, it doesn't happen in the lower organisms. Clear?
Yes, ma'am.
Then comes the Golgi apparatus. Now, the Golgi apparatus, this was actually found or discovered or described by a person called as Camillo Golgi. And since it was described by him, so it was described by him, so it is named after him only, Golgi apparatus. Now, Golgi apparatus is also a membrane-bound cell organelle, and it is also made up of a lot of cisterns, right? The infoldings that are there. Yes, what happened?
Nothing, ma'am.
Are you smiling?
Ma'am, because the name is funny. Camila Cool.
Okay. Camillo Golgi, yes. See, he named an organelle after him.
Yes,
Imagine.
ma'am.
Imagine there would be Umar Golgi. But you have to discover something to be named after, isn't it? So you
Yes,
do discover something someday, inshallah. Okay. So the Camilla Golgi, it is a membrane-bound cell organelle. So you are only going to see it in the case of eukaryotes and not in the case of prokaryotes. Talking about the functions of the Golgi apparatus. So it is responsible for the packaging and the dispatching. So it's like whatever materials are synthesized by the endoplasmic reticulum. For example, they have synthesized proteins, they have synthesized lipids, right? So now where will it get packaged in? So it will get packaged in the Golgi apparatus, and then it will be dispatched by the Golgi apparatus. So imagine the Golgi apparatus like a delivery center, how the Amazon and stuff delivery centers are there. So what they do, they put stuff in a box and then they send it to the correct address. So the packaging and the dispatching of the products that are synthesized by the endoplasmic reticulum to the targeted site, to wherever it is required. So there the Golgi apparatus will be functioning. Other than that, it is responsible for storage, it is responsible for modification, for different kinds of packaging, and they are responsible for the formation of simple sugars from the complex sugars. And one of the cell organelles that we saw, which was lysosomes, they are also synthesized by the Golgi apparatus. Meaning, once the endoplasmic reticulum will release its product, it will be sent to the Golgi apparatus. Golgi apparatus will pack it and make it into a vesicle. And that vesicle is what we call as a lysosome. So like these three structures, they are connected. That is why we call them as the endomembrane system. Endoplasmic reticulum, vacuoles, lysosome, and Golgi apparatus. Four of these things become the endomembrane system. Clear?
yes, ma'am.
They're very connected to each other. And the Golgi apparatus is also found in the case of plants, but in the case of plants, you don't call it as Golgi apparatus, you call it as dictyosomes. Clear?
Yes, ma'am.
Any doubts up until now?
No, ma'am.
All right. See, I haven't asked you to write down anything in today's class because I have to complete your syllabus before your examination. But don't worry, these notes will be provided to you. Okay, then you can study from these notes.
Okay,
Okay. So do you have any doubts up until now?
no, ma'am.
Okay. So we'll keep it till here only for today's class and we'll continue in the next class. Okay.
Ma'am,
Hmm.
I think we are missing a topic.